Hey guys, it's Rani. I just recorded a longer form deep dive into gene set enrichment analysis, or GSEA, which I'll link above. But I wanted to also record a really quick bite-sized tutorial and address some of the most common questions that we hear about gene set enrichment analysis. So what I'm showing here on the Pluto canvas right now is a differentially expressed gene list visualized as a volcano plot. So when you do differential expression analysis, we're looking at differences in expression across all genes that were measured in the genome in this particular comparison. So here I'm comparing ethanol treatment versus no ethanol, and you can see that a number of genes changed significantly, up here shown in red or down shown in blue. And what we're trying to address with pathway analysis is really asking, okay, so these genes, changed individually. What kinds of pathways are they participating in, more broadly speaking, so that I can understand the biological meaning behind these changes? So to do that, we can use something like gene set enrichment analysis, or GSEA, which produces these characteristic enrichment plots. And really quickly, this heat map along the x-axis of the enrichment plot is showing similar kind of information as what's on the volcano plot, where we have our genes ranked from left to right here along the x-axis. Genes that increased in expression based on their log fold change are shown in red, and genes that decreased are shown here in blue. So imagine you've got all of your genes here ranked by their log2 fold change. Every time we see one of these black tick marks, we're finding a gene that was annotated in the gene set that we're looking at. So this particular study was interested in cholesterol-related genes, so we're looking at the hallmark cholesterol homeostasis gene set here. And we can see that indeed a couple of those cholesterol homeostasis-related genes move in an upregulated and coordinated fashion here in the comparison that we were looking at. To help make this a little bit more real, what I can do is highlight those cholesterol-related genes on the volcano plot here. So I have the ability to save and bookmark gene sets or gene lists, and so I can select one here in Pluto for this cholesterol homeostasis and save changes. And now what I can see is those cholesterol-related genes highlighted on the volcano plot. To kind of compare with the batch here that I'm seeing upregulated on the GSEA enrichment plot. So hopefully that helps with the kind of basic understanding of gene set enrichment analysis, really what it's doing, what it's looking for here, how it's pulling out those different genes that were annotated. Of course, that's just visualizing one gene set at a time, so cholesterol homeostasis. We can also visualize pathway analyses as a bar plot where we're really looking at enrichment scores across all gene sets and lots more. So feel free to dive into that longer form video that I had recorded, but hopefully this was just a really bite-sized overview to get you started. Thanks.